Hi, I'm Aditya Bardia. I'm a breast medical oncologist at Massachusetts General Hospital. So my talk was on liquid biopsies and genotype-directed therapies in metastatic breast cancer. The first question to consider is, should women with metastatic breast cancer receive routine tumor genotyping? If we look at the management of metastatic breast cancer, we have multiple therapeutic options: endocrine therapy, chemotherapy, clinical trials. So the idea is, for an individual, can we identify actionable mutations and then based on that select therapy? For example, a patient with metastatic hormone receptor-positive breast cancer, if you sequence the tumor and identify an actionable alteration such as an ESR1 mutation, that could be of potential clinical utility. ESR1 mutations essentially make a tumor estrogen independent. They are still dependent on the ER pathway, but they become estrogen independent. So if you use a therapy that lowers estrogen, like aromatase inhibitors, those therapies are unlikely to work. And essentially, ESR1 mutations represent a mechanism of acquired resistance to aromatase inhibitors. But such tumors can still be inhibited by estrogen receptor blockers or selective estrogen receptor degraders, or SERDs like we like to call them. There are a number of oral SERDs in clinical development, an example being elacestrant or RAD1901, which in phase 1 clinical trial demonstrated pretty impressive clinical activity for patients with metastatic hormone receptor-positive breast cancer, including the ones with ESR1 mutations. And there's an ongoing phase 3 clinical trial looking at activity of elacestrant versus standard of care endocrine therapy for hormone receptor-positive metastatic breast cancer. And a co-primary endpoint of that trial is to specifically look at ESR1 mutant breast cancer and look at the differential activity between elacestrant and standard endocrine therapy. So that's an example where, based on tumor genotyping, you can select an appropriate targeted therapy. In terms of a potential challenge, the challenge is clonal heterogeneity. When we sequence tumors, be it tissue or plasma, there are a number of molecular alterations that can be seen at different mutant allelic fractions. And this is particularly true for metastatic disease, for a tumor that has received a number of therapies. As the tumor evolves, it gains additional mutations, and that results in substantial clonal heterogeneity. So how do we target a tumor that has clonal heterogeneity, has multiple driver mutations? There's no easy answer to this question. There are a number of different approaches that are being used. Combination therapy is one approach to consider. Use of antibody drug conjugates that essentially target lineage-specific or cancer lineage-specific antigens, and use therapies or antibodies that target those antigens to deliver higher doses of chemotherapy and optimize the efficacy-toxicity ratio is one such approach. And a third approach to consider is patient-derived models. For an individual patient, based on blood or tissue, you actually generate patient-derived xenografts, and then in that xenograft, test for drug combinations and then bring it back to the patient. And finally, for precision medicine, I think it needs to move towards early breast cancer. So over the years, we have utilized precision medicine in metastatic breast cancer, but majority of breast cancer is localized breast cancer. So potentially, one could consider precision medicine approaches for early breast cancer as well. That would have a substantial impact for a large number of patients with breast cancer. The challenge has been that it's very difficult to detect small amount of either circulating tumor DNA or circulating tumor cells. So from a technology perspective, it's been very difficult to implement precision medicine approaches in early breast cancer. We are doing ongoing trials in the minimal residual disease setting, and the idea is that with those trials, we would be able to implement the precision medicine vision in early breast cancer as well.